Biology CPT Part 3: Molecular Genetics. David now has another problem. His muscles are cramping because of his strenuous exercise. Sandy, his coach, tells him that his muscles are tearing and will be rebuilt, and suggests that he consume some protein to help the process. David is shocked at first and wonders why the amino acids in his protein powder help build muscle. We will now be going in depth of how muscle buildup occurs. Muscle cells contain a large amount of protein, which is built through a process called protein synthesis. In this process, amino acids are used to produce complex protein structures. What are amino acids? Amino acids are the structural units that proteins are made of. They are molecules which contain. Following is a flowchart that explains the process of protein synthesis. It may look a little big at first, but we will be going in depth into each and every process that occurs. Protein synthesis occurs in two steps. The first is called transcription, and the second is called translation. The first part of protein synthesis is called transcription. In this process, promoter regions in the DNA which are rich in adenine and thymine molecules are scanned and recognized by the enzyme called RNA polymerase. These promoter regions only have double bonds, which means they are easier to break. As soon as transcription begins, the 5' cap is added to one end. The RNA polymerase binds to these promoter regions and then nucleotides are rapidly added to these regions using this enzyme. Once the enzyme encounters a specific termination sequence, the transcribed molecule gets a 3' poly A tail and is then released. Before the mRNA can be released into the cytoplasm, it must undergo another process. This is called mRNA splicing. In this process, pre-mRNA introns are cut out by the spliceosome, which then fuses the exons back together. This is essential because it makes sure that the mRNA will be read correctly in the next step. The mRNA is then traveled outside of the nucleus into the cytoplasm. The next process is called translation. Firstly, ribosomes bind to the mRNA by recognizing the 5' cap of the molecule. Another molecule called transfer RNA carries an amino acid called methionine. This molecule also carries the anticodon UAC and is the initiator transfer RNA. This binds with the small ribosomal subunit forming a complex. The complex then scans through the mRNA until it finds the initiator codon. The large ribosomal subunit then bonds to this complex and forms a larger complex, sort of sandwiching the mRNA in the middle. Next, the complex has two binding sites, called the peptide site and the acceptor site. The initiator tRNA binds to the peptide site, leaving the acceptor site empty. Another tRNA molecule brings other amino acids according to the code of the mRNA, and peptide bonds are formed using an enzyme called peptide transferase. Translocation also occurs, where the ribosome moves along the mRNA molecule while scanning it, bringing more amino acids forming a polypeptide chain. Finally, once the ribosomal complex scans a stop code, a release factor protein dismantles the complex. The subunits separate and the polypeptide chain is released. The polypeptide chain that is released is inactive and useless at first. The chain is modified and amino acids and sugars are added and deleted in the Golgi complex of the cell. The functional groups of the polypeptide chain interact, and the polypeptide chain may interact with other chains to produce quaternary proteins. These chains then form a fully functional and large protein. Branched chain amino acids, or BCAAs, like the one in David's protein smoothie, are a group of 3 essential amino acids that we can only get from our diets. These include leucine, isoleucine, and valine. These amino acids are vital to building muscle, decreasing muscle fatigue, and helping with muscle soreness.